established body bank and database dedicated for cancer genome research. Today she will talk about the findings. The title of the third talk is Dynamic Genomic and Cellular Alterations in Multi-Step Cancer Development. Let's welcome Chen.
Good afternoon, everyone. Thank you so much for self-press invitation. And this is my first time in West Lake University, and I just see my student. She got my master degree, and she will get her PhD degree next year from this university. It's fantastic. Today I will talk about the cancer initiation and evolution part, and I know that everyone knows that cancer is have a relationship with the aging, so that might be the reason that you invite me to give a talk here. During the last 10 years, I have been focusing on this type of cancer, esophageal squamous cell carcinoma. But according to pathology classification, there are two types of esophageal cancer. One is the esophageal adenoma, and the other one is the esophageal squamous cell carcinoma. And more than 50% of the SCC were identified in China. So we have our institute and our National Cancer Center have been studying this type of cancer for more than 60 years. We think, like other types of cancer, and the esophageal cancer includes the normal stage and the two pre-cancer stage. And cancer carcinoma in situ and invasive carcinoma. Why we chose this type of cancer to study the cancer initiation and evolution? Because not like other types of cancer, the epithelium of this esophageal squamous epithelia is very simple. It includes 3 layers: the basal layer, and above the basal layer there are 25 layers and upper squamous. So we could guess these epithelia use the endoscopy, and in our hospital we could have the biopsy and sent to the pathology department and to see if the tissues are normal or pre-cancer tissue or the carcinoma. So, and we think that is really good because we could get samples from different stages. And another point is we could see the whole multi-step stage of this during the carcinogenesis. So my lab has been focusing to the dynamic evolution of this type of cancer and using the multi-stage omic platform. And at first, we used the GWAS and to see the total variation associated with this type of cancer in the high incidence and the sporadic areas in different parts of China. And we also identified that the gain-of-rapid infections, like the 2 risk SNPs in the alcohol 2G enzyme in the alcohol metabolism pathway are associated with risk of ESCC. But we know that not all the germ variation mutations are very important in cancer initiation. But there is one knowledge gap is how we define the normal epithelia. We could see, so we need to define that the aged normal epithelial cells. We have 5 donors, and the average of these 5 donors was 87 years old, and the average donor we got 9 organs. Take the esophagus epithelium as an example, and we just cross the epithelial layer and into 5 layers, continuous layer, and on each layer we got 10 mini-blocks. So only this thin epithelia tissue, we got 50 mini-blocks, and we could have the 3D construction of this epithelia. And for other 8 organs, we also have the similar construction. So we use the laser capture macro dissection, and each mini-box have the 600 cells. And in all, we have 737 mini-box, and with 3D spatial location for WES, whole genome, and for exome sequencing. And we could see that the somatic mutations and the cognitive variations with these normal epithelia tissues. In all, we got the 53 unique SNPs and 444 alleles. And we could see that the distributions of these various allele frequencies of the mini-box, and it could suggest that the various degrees of clonal expansion in different organs. And it's very interesting, we could also identify the copy number variation events in the normal epithelia. Like the esophagus epithelia, we could see that the arm level copy number variation occurs from 3, 5, and 7. These three amplifications could be identified in the esophageal carcinoma. But in this normal epithelia, age to normal epithelia of the esophagus, we still could identify in some individuals. And then we do the pan tissue analysis, and we identify that the cancer driver genes also could be identified in these normal tissues. Like we are very familiar with the Nortran TP53 mutations were reached in these normal esophagus epithelia. More than 74% of mini-box carrying more than one driver mutations. And then we do the correlation analysis between the mutant clone size and the mutation burden. We could see two different scenarios. One is this part, and these clones are larger, like the esophagus and the cardia. But these clones without the microstructure, but this large mutant clone expands to microscopic that is less mutations. But like clones in the colon and the rectum, and these clones with microstructure, and these clones are very competitive, and it might evolve independently during aging. And then we do the polygenic gene analysis and reconstruct the spatial clonal expansion. And take the PN9 donor as an example. You could have the 49 millibox from the esophagus, and we could see 8 clones. Let's see the clone. And these 8 clones could expand to microscopic scale, nearly 1 micrometers. And we then want to see what mutations, the mutations of number drive this clone expansion. You could see some clones like the clone 1 and clone 2. These 2 clones were driven by the NOT1 and TP53 mutations. But we first see that some clones in the normal epithelia cells could be driven by the amplification of the arm level chromosomes 3, 5, and 7. So that means that different clones might have diverse clonal genome-driven patterns. So today I will only focus on the esophagus, but if you are interested in other organs, how clone expansion in other organs, we will talk during the conference. So the first time to construct the whole body map of somatic mutagenesis in the morphologically normal human tissues. And this study reshaped our understanding of clonal formation and expansion in normal human tissues. And we also want to see the mutational signature in normal tissues. Because the average of the 5 donors is 87 years old, we could see that the aging-related signature, SBS1 and SBS1 and SBS5, and are very widely present in multiple organs. The light blue indicate and the dark blue indicates the aging-related signature. So we think that maybe because the indication of this study, because we only have the donors aged old from the old people. So next time we want to see if we could have the young people and to see the how clone expansion during the aging process. So first, I want to just mention the whole genome sequencing of the cancer. Because the somatic mutation also has the consequence of the complex etiology, and we could use the SBS, the genomic fingerprint, to guess against the etiology of the patients. So not about, only about the aging, we could see the environmental factors and the intrinsic factors, endogenous aspect, the A2A, apoptotic genesis is predominant contributes to A2A upregulation and enhanced immune infiltration in old patients. So we raised one question: if we could trace the dynamic change of the signatures during cancer development. We have a very large cohort. During the last 20 years, I followed this cohort in Yingxian, Henan province. This area is the esophagus instance of this area, maybe the highest in the world. And we use the endoscopy for screening. And during the last 15 years, and we could see that the early detection rate increased in this area, and the cancer incidence and the mortality decreased rapidly. But we can't copy this strategy in other areas because we need it's cost and it's invasive, and we need good endoscopies. So we hope that to identify the key molecular events driving molecular transition and to elucidate the clonal dynamic evolution during the SCC development in humans. So during this cohort, we could get the different epithelials and follow these individuals from the normal to precancer and the cancer stage. And in all, we got the normal epithelials at different ages. The number is 22. And we got the 9 LGM, the early precancer stage tissue, and the 7 HGM tissue is the late precancer stage, and the 4 cancerous tissues. And in all, we got the more samples and to see the clone expansion during cancer development. And this slide shows that we have the normal epithelial tissues all of the individuals at the different ages from 22 to 76. And we identified that the individuals aged less than 55 years old, the mutation rate is mutation, we could have the average mutations is 80.5. But if the individuals age more than 55 years old, the average of number of mutations is 33. But compare that to the different stage of the cancer, we could see that like the LGM and HGM and the cancer, we could see it increase rapidly. But for the copy number variation, displays no aging increasing trend. we could extremely rare in normal samples, but there is an exception, the donor age 3, and this individual has the gastroesophageal reflux, and we could see the sporadic copy number variations in chromosome 5, 7, 9, and 17. As for the LGN, the pre-cancer stage and the ESCC stage, we could see more copy number variation, and it increased significantly during cancer development. And for the frequency of the different mutations of the cancer-related genes, we see that the TP53 and NOTCH1 exhibit divergent trends during cancer development. We see that at the normal stage, we could see lots of mini-box at the NOTCH1 mutation. But from the LGN, the pre-cancer stage, and we can see lots of clones carrying TP53 mutation. And this shows that the TP53, and from the LGN stage, we can see that TP53 status is positively correlated with the copy number variations. And when we plot the clonal architecture based on the driver mutations and copy number, we can see that at the normal stage, the clones are very small, and it was driven by the NOTCH1 mutation. But for the clones at the pre-cancer stage and the cancer stage, we see that the clones are relatively larger, and it was driven by the TP53 mutation. So they are completely different. And we see that at the LGN, the TP53 mutation clones also carrying more copy number variations. When we see that the polyclonal architecture in normal esophagus, but this donor is the age is only 39 years old. We could see that the randomly rare mutations. But for this donor, age is 66 years old, and we could see the large clone, and it was driven by NOTCH1 mutation. And when we go to the LGN, the early pre-cancer stage, there are 4 clones from this donor, and there are the clone 1 and clone 2 are the LGN clone, and clone 3 and 4 are the normal clones at normal stage. And we could see that the clone 1 and the TP53 mutations and LOH, and we could see, and these clones also carrying lots of the gain and loss of copy number variations, and it's also on the arm level copy number variations. And we could see other clones from other the clones, the similar results from clones from the LGN stage. That means the LGN clones with TP53, lots of the LOH actually is higher number of copy number variations compared to those without the TP53 LOH. And for the clones at the HGN and the SCC, the clones are much similar. So it indicates that the LGN might be a good stage to do the early detection, because it's very easy for us oncologists to identify the pre-cancers, because use the endoscopy we could have these patients who just cut these lesions at the LGN, and the patients would go back home in after this surgery is only 30 minutes. But if the patients at the late stage, like the cancer, and they have to stay in the hospital for more than 1 week after the surgery, and they need other chemotherapy and immunotherapy. So this tell us that the pre-cancer stage is very important, and it is the genomic change might start at the early stage of pre-cancer stage, and the TP53 TP53 mutation and LOH might be the important marker. And then we do the CD53 biallelic loss and the chrome number variation and see that the chromosome instability. And we do the CRISPR-Cas9 screening of these chromosomes and hope that we will have novel results and to see which genes of the genetic or the genomics change to contribute to the cancer development. So this paper was tell us the genomic alteration relevant pre-cancer and to cancer agents and ESCC development. And except the genomic change, and we also focusing on the cellular alteration during cancer development. And benefits from this lab cohort, not like the other single cell study, and it's still focusing on the samples at the normal stage and the pre-cancer stage. Then, long story short, we could see that also that the different change of the epithelial cells and the fibroblasts and immune cells during the cancer development. And we identified that the mucosal defense and this program is very important, especially in the esophagus, because every day we have the water or the food, and it might damage our epithelia and mucosal of the esophagus. And the mucosal defense program is very important to protect our esophagus. And ALXA1 gene is the top gene we identified in this program. And it's the expression of this mucosal defense program is correlated with the esophageal squamous cell carcinoma patients' survival. So what's the function of this gene? And this gene, this protein could be expressed in multiple human organs, and the main function was reported as involved in anti-inflammatory effects, and it was separated into the extracellular space. And ALXA1 as a ligand is known to bind to the FTR family. And then we could see that during the ESCC development, and R level and the protein level of ALXA1 of the epithelial cells decreased during cancer development. And the expression of ALXA1 is negatively correlated with the cross-reactor scores. And based on the spatial transcriptomic, we will see that the ALXA1 suppression and the normal fibroblasts to cause the transformation both occur at the pre-cancer stage. And ALXA1 could bind the FTR at the fibroblast membrane and change the after the binding, and it could inhibit the ERK and AKT and SMAD3 signaling in normal fibroblasts. So during the cancer development and the expression decrease, and it might initiate the powerful transformation at the pre-cancer stage. So we could see that also in the mice and during the cancer development, and ALXA1 could reduce the clot formation and suppress the tumor development. So we could identify the genomic change and the cellular change at the pre-cancer stage. So it's very useful for us, for our oncologists, and especially the endoscopists, and to identify the early cancer patients. So but we still have no idea how to cut these lesions, because when we see the lesions endoscope, we just cut the epithelials. How to do that? So I think that spatial transcriptomics will us the good information and will help us to define that how large of the lesion area we will cut. So this is yeah. So we have a very good slide, and this slide consists of the whole stages of the epithelium, the normal stage and the LGN, HN, precancer stage, and the esophagus squamous cell carcinoma on the same slide. So we could see that the different cells are at the different positions and their stages. So, if we could see that in clinic, the lesions, and we know that how large we will cut and help the patients. So thank you so much. Sorry, I know time is limited, so I didn't talk more about the details. At the last, I will thank my colleagues and definitely thank the individuals and patients supporting our research. Thank you.
We have 2 minutes for questions.
Thank you, Professor Wu, for your fascinating presentation. Your pathological analysis of the genetic issue is very fascinating because I think it's one of a kind in this meeting till now, and I wonder if there's any possibility to relate the genetic differences between the clones and their spatial characteristics in this issue. And by doing this, I think we can know the different level of the changes of the genes through the malignancy of the cancer. Thank you.
So you mean the germline or somatic? Oh yeah. We hope that because we do the GWAS, so we might know